Did you know that without enzymes, the chemical reactions keeping you alive right now would take thousands of years to happen? Enzymes are the superheroes of our cells. Hello everyone, and welcome back to your channel name. In today's video, we are breaking down the enzyme-substrate interaction. We will look at exactly how these molecular machines work, what the active site is, and the secret behind the lock and key mechanism. If you are studying biology or biochemistry, this video is for you. Let us dive in. First, let us identify our main characters. On the left, we have the enzyme. This large blue structure is a protein. Its job is to act as a biological catalyst, meaning it speeds up reactions without getting used up itself. On the right, we have the substrate molecule. This orange shape is the reactant. It is the specific molecule that the enzyme is going to work on. Think of the enzyme as a factory machine, and the substrate as the raw material. Now, look at how they fit together. This is not random. The enzyme has a specific pocket called the active site pocket. Notice the shape. The substrate fits perfectly into this pocket. This is what we call three-dimensional shape complementarity. Scientists often call this the lock and key model. The enzyme is the lock, and the substrate is the only key that fits. This ensures that enzymes are highly specific. An enzyme for digesting sugar will not accidentally digest proteins. Let us zoom into that pocket. It is not just an empty hole. It is a chemical workshop. The binding site is the area responsible for holding the substrate in place through temporary bonds. The catalytic residues, you see those glowing yellow dots? These are specific amino acids. Once the substrate binds, these residues get to work. They might stretch the substrate bonds or provide the perfect environment to break them. This lowers the activation energy, making the reaction happen much faster. To summarize the definitions on the screen, the active site is the specific region where binding and catalysis occur. The substrate is the reactant that binds to the enzyme. And remember, this binding is temporary. The substrate does not live there. It visits, gets changed, and leaves. Finally, let us look at the equation at the bottom, which tells the story of the reaction. Enzyme plus substrate forms the enzyme-substrate complex, which then becomes enzyme plus product. First, the enzyme meets the substrate. Second, they bind to form the enzyme-substrate complex. This is the moment the reaction happens. Third, the substrate turns into the product. Crucial point. Look at the enzyme at the end. The enzyme is released unchanged. It is ready to grab another substrate immediately. One enzyme can process millions of substrate molecules. Before we wrap up, here is a bonus point for your exams. Since enzymes are proteins, their shape is delicate. If the temperature gets too high or the pH level changes too much, the enzyme can lose its shape. This process is called denaturation. If the shape changes, the substrate will not fit anymore, and the enzyme stops working. So, to recap, enzymes are specific, reusable catalysts that use their active site to turn substrates into products. I hope this diagram makes sense now. If you found this video helpful, please hit that like button and subscribe to the channel for more biology breakdowns. Got a question about enzymes? Drop it in the comments below, and I will answer it. Thanks for watching, and I will see you in the next video. Highly specific, enzymes are picky. One enzyme usually works with only one specific substrate. 3D shape complementarity. The shape of the active site matches the shape of the substrate perfectly, like a puzzle piece. Temporary binding. The substrate doesn't stay there forever, it binds, reacts, and then leaves. Catalytic function. The enzyme lowers the energy needed for the reaction to happen.